All right, let's dive right in. Today, we're shrinking down to the molecular level to talk about one of the coolest tools in all of biotechnology, restriction digestion. Think of them as tiny, programmable scissors that let scientists edit the very code of life. It's pretty amazing stuff.
So, to get where these molecular scissors came from, you have to picture this ancient, microscopic war that's been going on for billions of years. Imagine you're a single bacterium, and you're getting attacked by a virus. You don't have an immune system like we do, so how on earth do you fight back?
Well, it turns out bacteria have a secret weapon, something called a restriction enzyme. It's this brilliant little protein that acts like a security system. When a virus injects its DNA, the enzyme scans it, recognizes it as a foreign invader, and just chops it to pieces. Threat neutralized. It's a seriously elegant and effective way to defend yourself. But here's the really incredible part. How do these enzymes know exactly where to cut? They aren't just snipping randomly. Their precision is what makes them so powerful. So let's get into how they find their target. And it all starts with how they get their very specific names.
Okay, so you see a name like EcoR, and it probably looks like a bunch of scientific gobbledygook, right? But it's not. It's actually a code that tells you the enzyme's entire life story, where it came from, what organism it was found in, the whole shebang.
Let's just break this one down. The E stands for its genus, Escherichia. The Co stands for its species, coli. Yep, that's the famous E. coli. The R tells us the exact strain it was isolated from, in this case, RY13. And that Roman numeral I at the end, that just means it was the very first restriction enzyme they found in that strain. See? It's like a perfect little scientific address.
So now we know the enzyme's name, but what is it actually looking for in the DNA? It's searching for something really specific, a special sequence called a palindrome. Now, a DNA palindrome is a little different than a word palindrome. It's got this cool two-sided symmetry.
This makes it super clear. We all know words like race car, right? Reads the same forwards and backwards. Well, a DNA palindrome does the same thing but across the two strands of the double helix. If you read the top strand from left to right, or 5' to 3', it says GAATT. And if you read the bottom strand, its complementary partner, also from 5' to 3', it also reads GAATT. That perfect mirror image is the signal the enzyme is looking for.
So the enzyme finds its palindromic target. Awesome, but the next big question is, how does it actually make the cut? Because, you see, not all cuts are the same. The way the DNA is sliced creates totally different kinds of ends, and each one is useful for different things in the lab.
Let's start with the first and maybe most common type of cut. This one creates what we call a 5' overhang. Basically, the enzyme makes a staggered cut across the two DNA strands, which leaves this little single-stranded tail. And that tail is called a sticky end for a really good reason. It has these exposed DNA bases that are just eager to find a matching partner and stick to it.
Look at our friend EcoRI again. You can see it cuts right between the G and the A on both strands. That staggered slice leaves a little AATT tail hanging off each piece. That's your 5' sticky end, and it is absolutely perfect for when scientists want to paste different pieces of DNA together, almost like molecular Velcro.
And it just makes sense. If you can have an overhang on the 5' end, you can have one on the 3' end too. It's the same idea. A staggered cut that creates a sticky end, but this time the tail is just on the other side of the strand. An enzyme called PSTI is a perfect example of this. It cuts after the A in its recognition sequence, and that leaves the overhang on the 3' end. At the end of the day, both 5' and 3' overhangs give you those super useful sticky ends that are the bread and butter of genetic engineering. But what if you don't actually want a sticky overhang? What if you just want a nice, clean break? Well, that brings us to the third type of cut. This one creates what scientists call blunt ends.
So an enzyme like small I is a blunt cutter. It finds its target sequence, CCGG, and just slices straight through the middle. No overhang, no sticky tail, nothing. Just two perfectly flat or blunt ends. These are really useful too, just for different kinds of lab techniques where you need a straight edge.
Okay, so we know how these enzymes find their target and how they make the cut, but to actually use them in a lab, you have to give them the perfect working environment. Seriously, it's like baking. If you don't get the ingredients and the temperature just right, the whole thing will fail. And the single most important factor is probably temperature. Since most of these enzymes evolved inside bacteria, they do their best work at 37 degrees Celsius.
which, yep, is basically human body temperature. If it's too cold, they get lazy and slow down. If it's too hot, they fall apart and stop working altogether. So 37 degrees is the sweet spot.
But it's not just about the heat. Your DNA has to be super pure, with no junk like salts or ethanol left over. You also have to add just the right amount of enzyme. Too little won't get the job done, but believe it or not, too much can cause huge problems. Even the total volume of your little reaction tube matters.
So this is what a standard recipe might look like in the lab. You've got your water, a special buffer solution to keep the chemistry happy, and your DNA. But there's a golden rule every biologist knows. You always add the enzyme last. You get everything else mixed together perfectly. Take a deep breath, and then you add your molecular scissors to kick off the reaction.
And following that recipe is absolutely critical. Because if you mess up the conditions, if you get lazy, the enzyme can go completely rogue. And that leads to a really common problem that can totally ruin your whole experiment.
This problem has a great name: star activity. It's what happens when the enzyme gets sloppy and loses its famous precision. Instead of only cutting its one perfect target site, it starts cutting at other, kind of similar sequences all over the place. Your precision molecular scissors have just turned into a pair of rusty hedge trimmers.
And you can see here, pretty much any deviation from that perfect recipe can cause trouble. Using the wrong buffer, leaving it to incubate for way too long, or adding way too much enzyme. For instance, enzymes are stored in glycerol to keep them stable, so if you add too much enzyme, you also add too much glycerol, which throws off the whole reaction. The takeaway here is really simple. You have to follow the recipe to the letter.
I mean, think about it. The journey of these enzymes is just incredible. From a simple bacterial defense mechanism to the absolute cornerstone of the entire biotech revolution. These molecular scissors gave us the power to read, edit, and rewrite the code of life itself, which led to new medicines, new crops, everything. So the real question isn't what they've already done for us, but what will we build with them next?